Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we are going to learn about non-coding RNA. But before we start learning about non-coding RNA, let's do a quick recap of what we have learned till now. So, when we started with epigenetics series, we first learned about what is epigenetics, what do we study in epigenetics, and how this field was emerged. So in epigenetics, we study all of the changes in the DNA which are heritable, means they can be passed down from the parent generation to the daughter generation. But those changes in DNA are not sequence specific, means those changes are not in the DNA sequence itself, but they are on the DNA. So those changes which are done on the DNA without changing its sequence. And again, it is interesting because it can be passed down from generation to generation. So this idea was rejected when it was proposed by Lamarck. So I hope you remember theory of Lamarckism, which was rejected at that point of time, which suggested that acquired traits can be passed down from generation to generations. But then this theory was supported by epigenetics because in epigenetics, all we do is to study about those changes that happen on the DNA, like on the genome. That is why the name epigenetics on the genome. But those whatever those changes are, they are not in the DNA sequence. Means the sequence is kept the same, but the way that DNA is read by the cell, it is differed from the cell to the daughter cell, or in the single cell, but in its different life stages. For example, if the cell is in pluripotency stage, it will have the same DNA as that of the cell when it becomes more differentiated, right? But in both of the stages, some gene products are more in the pluripotency stage, or some gene products are more in the more differentiated stage. So how this is achieved? So this is achieved by the epigenetic factors. So what makes the DNA different? So DNA is more like a script. So this idea was proposed by the researchers after a series of experiments, after a lot of case studies. We also learned about one case study, Dutch hunger winter, in our first lecture. So through the years, through the decades, scientists got to know that DNA is not like a template. It is more like a script. So script can be read by different actors differently to give the different acting, right? So like the product will be different from the whatever was given in the script, depending on which actor is going to read that script. It is the same way. The DNA is like a script which can be read differently by different cells, or it can be read differently by a single cell which is in its different life stage. Okay, for example, let's assume that there are two twins. Though both of them have same set of DNA, but still they differ. So they differ because their DNA is same, but that DNA is read differently by both of them, and that difference is due to either the environment they are living in, or the choices they are making, their lifestyle. All of those affect the genome, and that is what we study in epigenetics. So the changing the factor that is making the change in the DNA, something which is different in that DNA, is not the sequence but the epigenetic factors. The way epigenetics factors are placed on the DNA, that is differed from the cell to cell, or from the in the cell in its different stages of life. So this is so that's why for studying epigenetic factors, first we need to brush up our basics, right? So we learned about chromatin, how the DNA is packaged in the cell. So DNA is quite long, and this long stretch of DNA can be packaged in 6 micrometer, and I mean that small of the cell, and that is done by the packaging. So this is a next level of packaging that is done in the cells. So we learned about that. After learning about packaging, we moved on to the modifications. So epigenetic factors are actually marks, epigenetic marks. Those are the chemical modifications that are done on the chromatin. So chromatin consists of the DNA as well as the proteins around which the DNA is packaged. Those proteins are histones, which are positively charged. Because they are positively charged because they have a lot of basic residues, basic amino acid residues, which give them positive charge. So they are positively charged, and that's why the negatively charged DNA can be wrapped up around histones. So in the chromatin modifications, we first learned about DNA modifications. So modifications are nothing else but the addition of certain chemical moiety, certain chemical group on the DNA or on the histones. So in the DNA modifications, we learned about DNA methylation, DNA methylation being the most important one. So in DNA methylation, obviously we add CH3 group on the fifth carbon of cytosine of the CpG dinucleotide, right? So this DNA methylation, this process is carried out by the enzymes. So we learned about those enzymes which are known as DNMTs in detail, the DNMT3A, DNMT3B, DNMT1. Okay, so we learned about all of those DNMT. Again, there is one enzyme which transfers the methyl group on the tRNA, which is now known as TRDMT1. Okay, so we learned about all those enzymes in detail. Then we learned about DNA demethylation as well. So in the DNA demethylation, we learned about the different methods by which the methyl group can be removed from the DNA, in which there was an enzyme coupled process, means it means it involved the enzymes, and those enzymes were TET enzymes, the 10-11 translocation enzyme. So those are involved in DNA demethylation. So we this is all that we covered in DNA modifications. Then we moved on to the histone modifications. In the histone modification, we covered histone modifications recently, and in those series we learned about the histone methylation, histone acetylation, histone phosphorylation, histone ubiquitination, and histone tail clipping. So all of those different modifications, in the four of them, we add different groups on the histone. The histone N-terminal tails, or it can be added on the core amino acid residues as well. Then in the histone tail clipping, we cleave the N-terminal tail of the histones. So again, those histone modifications, depending on the location, depending on which enzyme is adding and what they are doing, we learned about the mechanisms of their working in detail. Again, we also learned about which of the marks are active mark, which of the marks are repressive mark, what are the enzymes which add those marks, what are the enzymes which remove those marks. Means we learned about writer enzymes as well as eraser enzymes. And after learning all of this, we are pretty clear with our basics of the epigenetics. So now we are going to learn about a new topic, and that is non-coding RNA. Okay, so in the cell, okay, I hope you all of you know about central dogma of life. So in the cell, DNA is fated to become RNA. I mean, DNA is fated to become to be transcribed and become RNA. Then RNA is fated to be translated and make protein. Okay, so DNA has to be read and it has to be transcribed to make RNA. Then RNA has to be read and it has to be translated to make protein. So this is the central dogma of life. But what if something goes wrong? Okay, in this process, there was observed that in the cell, 98% of the RNA was non-coding, means 98% of RNA did not code for any protein. So researchers thought that something went wrong, and this maybe something went wrong, but 98% non-coding RNA is not a joke, right? So they thought that the DNA could be junk DNA. Okay, but then it got ruled out due to the inventions that were made by the researchers, and they got to know that this non-coding RNA, they indeed have some functions as well as some regulatory role in the cell. So we were no till now we only have been discussing about the 2% of the RNA which codes for the protein. Indeed, that is the mRNA, the messenger RNA. But we also know a certain non-coding RNAs. I hope you know you have been learning about those. The tRNA, right? The rRNA, ribosomal RNA. So those are non-coding RNA, but they have quite important role in the cell. They are involved in protein synthesis, right? So we know about those, but we are going to learn about the non-coding RNA, which are involved in the epigenetic regulation. So first, let's categorize non-coding RNA. Okay, let's categorize non-coding RNA. Non-coding RNA is also written as ncRNA. So those are categorized on the basis of their role. So some has the functional role, major functional role in the cell, while some has regulatory role in the cell. Okay, so those which have a functional role, those are tRNA that we now discuss, transfer RNA, rRNA, the ribosomal RNA. Okay, so those are the functional non-coding RNA. But there are certain RNAs, non-coding RNA, which has regulatory role and they are mainly involved in the epigenetic regulation. So those are microRNA, piRNA, as well as long non-coding RNA, which we write as LNC RNA. So the microRNA, I am sure you have heard of it. So this micro miRNA are microRNA, okay. But we are not going to cover microRNA in detail in epigenetics series. Maybe we'll cover this in molecular biology series because it will be more appropriate to learn about it there. Again, the piRNA, those are known as PV interacting RNA because they interact with PV proteins. Okay. And long non-coding RNAs are known as long because they are long in the length, right? So we are going to learn about piRNA as well as long non-coding RNA in detail. So let's learn about piRNA first. Okay, yeah, one more thing. So those microRNA and piRNA, both of those are small non-coding RNA because they are small in size. The microRNA, they are about 19 to 24 nucleotide long, while the piRNA, those are 24 to 35 nucleotide long. Okay, while long non-coding RNA, they are obviously long, and that is why the name long non-coding RNA, and they are more than 200 nucleotide long. Okay, so this is also based on the size. Okay, yeah. So we were going to learn about PV interacting RNA or piRNA. So those, unlike, so those are also those are also involved in post transcriptional silencing, just like the microRNA, but the mechanism is different, and they are involved in post transcriptional silencing of transposons. Okay, so the microRNA, they are also involved in post transcriptional silencing, but the mechanism is quite different. For example, the microRNA, they are synthesized first in the primary microRNA. Okay, wait. There are processes that they are synthesized as primary microRNA in the nucleus. In the nucleus, they are on those the DROSA enzyme works, and then they are processed and converted into pre-mRNA, right? And then those are transported to the cytoplasm. On the cytoplasm, the dicer enzyme works. So this is a long processing of the microRNA. Okay, so this is how microRNA are processed. They are and then they can lead to the mRNA degradation, or they can lead to the post-transcriptional silencing. But the mechanism is quite different. While the PV interacting RNA, they only silence the transposon elements. There are certain non-repetitive elements that can be silenced by piRNAs, but that is not the main focus of the lecture. Today we are just discussing about how they are transcribed from transposon itself, and they silence the transposons. So they are transcribed from the okay, yeah. They are transcribed from the piRNA clusters, which are found in the transposons. So the transposons could have either unidirectional promoter or bidirectional promoter. Okay, so those piRNA, though they are synthesized in nucleus, okay, but the processing of piRNA, like the final working form of the piRNA, that is done in the cytoplasm. Okay, so what happens is that this is nucleus. and this is cytoplasm. Okay, those are transposons. Okay, they could be unidirectional or they could be bidirectional. Okay, so then from those they are transcribed and they form piRNA. Okay, and then they are exported into the cytoplasm and now they undergo the processing. So the first is primary processing. In this, they only get shortened. Okay, this is just shortening of the formed piRNAs. Okay, after that there is secondary processing. In the secondary processing, those shortened piRNA they undergo a cycle known as ping pong amplification. Okay, I'm not gonna discuss the ping pong amplification in detail in this lecture, but if you want, I can make a separate lecture for ping pong amplification process. Okay, so they undergo ping pong amplification, and that is how they are finally processed, and now they can go and silence transposon. Okay, so this is how piRNA are involved in transposon silencing. Now we will discuss about long non-coding RNA. Okay. So we are going to just discuss basics in the today's lecture, but and we will discuss about the examples such as exist, hot air in the next lecture. Okay. So long non-coding RNA being long, they are more than 200 nucleotide long. Okay, so long non-coding RNA have quite important role in taking the chromatin remodeling machinery and assigning that chromatin remodeling machinery to DNA. And again, they are involved in X chromosome inactivation. The exist one is involved in X chromosome inactivation. Again, they are involved in the silencing of Hox genes, the hot air one. Okay, so we will let's do the thing now that let's discuss about long non-coding RNA in the next lecture itself. So let's end the lecture here. Till then, see you. Bye bye.